Good morning everybody. In today's class I am going to discuss with you the second topic, sexual reproduction in flowering plant. So the flower is the site of sexual reproduction in all angiosperm. And flower usually has four valves. Outer two valves are called the accessory valves, which are not taking part in the sexual reproduction. Example, calyx and corolla. Inner two valves are called the essential valves, which are taking part in sexual reproduction. And example for essential valves are androecium and gynoecium. We usually call androecium is the male reproductive structure of a flower, whereas gynoecium is said to be female reproductive structure of a flower. Let us see the structure of a androecium. Units of androecium are called the stamens, and androecium consists of whorls of stem. Each stamen is made by two parts. A long, thin structure called the filament, and in terminal region there is a bulged structure called the bilobed structure called the anthers. The proximal end of this stamen is attached to the thalamus or the petal of a flower. The number and length of the stamens are different in different species flowers. Usually anthers are dithecous. The meaning of dithecous is when an anther is made by two lobes, such anther is called a dithecous. So here I am going to draw the internal structure of the anther. The anther is the tetragonal in shape, which has four sac-like structure in its core. And these sac-like structures are called the microsporangia. And these microsporangia, when they later develops and forms the pollen sacs. The structure of microsporangia. The outline of this microsporangia is circular, and which is covered with four layers. The name of these four layers are, it starts from outside to inside, epidermis, endothecium, middle layer, and innermost is called the tapetum. The outer three layers are for forming protection, and they also help in the dehiscence of the anther to release the pollen grains. But innermost layer is called the tapetum, which main function is to nourish the developing pollen grains. Each cell of the tapetum is large, which has a dense cytoplasma and also has the more than one nucleus. When anther is young, there is a mass of homogeneously spreaded cells or tissue, and that we call the sporogenous tissue. When the sporogenous tissue become mature, each cell of sporogenous is considered as a pollen mother cell. And these pollen mother cell undergo meiotic division and forms the haploid pollen grains. And this process is called the microsporogenesis. The pollen mother cell, this is called the pollen mother cell. When it undergoes meiosis, meiotic division, it produces a tetrad. And each tetrad will later become the pollen grain as soon as they dries up. The structure of a pollen grain. Pollen grain is a haploid male gamete. Pollen grain is a haploid male gamete. Each pollen grain measures 25 to 50 micrometers. The outermost layer of the pollen grain is called the exine and inner one is called the intern. The exine is not smooth, whereas intern is smooth. The exine is deposited with a hard substance called sporopollen, which is not usually hydrolyzed by any chemicals or alkali or acids. So that's why pollen grains can be stored for a longer time. Here and there on the exine there are small openings or the regions where sporopollen is not deposited. That portion is called the germ pore. On the outermost layer is called the exine. There are some regions which are not deposited with the sporopollen. These are called the germ pore. The inner layer, as we already know, this is called the intine, which is very smooth and it is made by cellulose and it is continuous. And inside the intine, there is a plasma membrane which covers the cytoplasm of the pollen grain. Inside the pollen grain, there are two cells. One is a large cell, another one is a small lens-shaped cells which is floating in the cytoplasm of the large cell. This large cell is called the vegetative cell and small cell is called the generative cell. This generative cell later divides into two male gametes. But vegetative cell will direct the pollen grain when enters the pollen tube when enters the style of the flower. So most of the cases pollen grains are released at only two cell stage, but only in few cases the pollen grains are released in 3 cell stage. So here I am going to conclude the structure of the stamen, microsporangia and pollen grain. In the next class we will continue with the female reproductive structure, gynosia. Thank you.